I would like to first thank the organizer for giving us this opportunity to present our work here. And I would like to thank you all for being here, especially on a Friday section. And it's a great conference. I enjoy every moment here. And yes, so we have a poster yesterday. So it's interferon alpha and interferon beta. So we want to recapitulate the disease state of the juvenile myositis. This is like the kind of work we're doing in our lab with the muscle system. We have we isolate the muscle myoblast from healthy patients or some myoblasts from the deceased donor. And then we try to model disease, or we try to recreate some rare disease in the healthy myoblast. So today it's part of the work. We try to create muscle fibrosis in a healthy donor muscle. So this is a part of a thesis work for Anadita, who recently graduated from our lab as a PhD student. Just a little bit background about the skeletal muscle fibrosis. It's a severe disruption to the muscle function, including the motile function and the contractile function with the muscles, and it increased the extracellular matrix, including the enhanced collagen deposition, and which increased the muscle stiffness as well. The cause of disease could be sustained and chronic inflammations, which results in accumulation of the fibrogenic growth factors, chemokines, and cytokines. Also, it could be arose from the disease, injuries, and sarcopenia. Picture on the right here, just showing with a normal muscle regeneration, the muscle will go through the normal phase, the inflammatory phase, the regenerative phase, and the remodeling phase, with a transient ECM deposition here. With the fibrotic muscles, it's different. It's with a progressive ECM deposition without the regenerative phase and the remodeling phase. In order to recreate this disease with our healthy myoblasts, we need an approach. The approach we chose is overexpression of the TGF-beta1. In many of the animal models, they have shown that with the TGF-beta1, the muscle fiber there's muscle fiber atrophies even in the absence of the physical injuries. And it reduces the contractile force with the muscles, and it results in the excessive ECM deposition, including the collagen and the fibronectin. These, which are exactly the phenotypes we're looking for. On the right side here, just shows with the TGF-beta1 overexpression, there's an excessive ECM deposition, fibrosis, and scar tissue formations, which this will lead to the incomplete recovery, the decrease in the muscle function and vascular perfusions, increase in pain, and likelihood of the re-injury. So with this approach, we still need a valid model system. Current model systems do not accurately recapture most of the states in the fibrotic human skeletal muscles. The prevailing system now in the research, including the 2D cell cultures, animal models, and the 3D cell culture. With the 2D cell culture, it's poorly modeled the physiological microenvironments, and the myobundles in there, they're on 2D, they are immature. There are great animal models to recapitulate the disease, but at the same time, there are interspecies difference, and there are poor predictors of the drug safety in humans and the drug efficacy in humans as well. And also, for the 3D cell cultures, it shows some hallmarks with the fibrosis, but did not reproduce the contractile muscles, and it did not evaluate the anti-fibrotic drugs. Here, I'm presenting the model system for our lab. So this is muscle myobundles based on a fibrin gel system. So we first mix the fibrinogens with the matrix gel in one tube, and the primary human skeletal myoblasts and the thrombin in the other tube. Right after we mix these two tubes, we quickly pour the gel onto a nylon frame on a PDMS mold. And then we keep them in a growth media for 4 days for the muscle cell to proliferate. And then we remove the frame from the PDMS mold, put them in a 12 well with the differentiation media, which induce the muscle cell to fuse together and forming the myobundles. And then after 7 days, they're mature. We can treat some drugs, chemicals, do some treatments to these myobundles for 4 to 7 days, and then we can perform functional testing. The muscle fibrosis is not only affecting the muscle, it also affects the nearby blood vessels. So it can form a physical barrier which impairs the vasculature, the blood flow, and the gas exchange in the nearby vessels. Also, the overexpression of TGF-beta1 could induce the endothelial cell mesenchymal transitions, and also it plays a key role in the atherosclerosis in terms of the general inflammatory. So here's just a picture showing the endothelial cells when we expose them with TGF-beta. There are loss of the EC markers, the cell-cell junction, CD31, and the vehicle headering, which is a CD144. And there's a gain of the mesenchymal markers, RFSMA, FSP1, vimentin, and the fibronectin. So to model the blood vessels, we have a blood vessel cistern, which is a collagen gel-based cistern. So we first mix the medial cells. We use smooth muscle cells and fibroblasts in the collagen, and then we pour the collagen into a custom-made chip, which is a 4-vessel cistern. And then after collagen gel, we endothelialize the inner wall of the blood vessels, and then we can perfuse this chip with our perfusion cistern with the peristaltic pump. So with the approach to recreate the disease and the cisterns we have 2 objectives in this study. The first one is to establish a fibrotic phenotype in a 3D human skeletal muscle cistern through overexpressing of the TGF-beta1. The second objective is to develop an integrated cistern with the muscles and the vessels to examine the functional changes due to the inflammations and the fibrosis. For the first goal, we follow the time frame to make the bundles, which is 4 days, with the growth media 7 days to mature, and we treat the bundles for 4 days with different doses of the TGF-beta1. And then we perform the endpoint testing at day 11. This will include the congenital passive force testing, gene expressions, and the immunofluorescence for proteins. We first look at ECM proteins, collagen 1, and we have 3 healthy donors, donor 1, 2, 3. As you can see here, in all 3 donors, once we treat them with TGF-beta1, the mRNA levels of the collagen 1 increase. We also confirm these results with immunostaining with the collagen. So as you can see here, with the 10 nanogram per mil of the TGF-beta, there's a high expression of the collagen 1 compared to controls, and we quantify it by measuring the mean fluorescent intensity, and you can see there's a significant increase once we treat the donors with the TGF-beta1. And then we look at the fibronectins, which is the same case. We saw significant increase in all 3 donors in terms of the mRNA level here, and also the protein level here, and it's quantified by the mean fluorescent intensities. So we isolate this primary myoblasts from a piece of biopsy. So there is certain amount of fibroblasts in the cell population. So the increased ECM productions could be due to the proliferation and increased amount of the fibroblasts. So we want to make sure it's not the case. So we check the vimentin markers, which is fibroblast markers. And for the control and TGF-beta, we can see the vimentin color is on the periphery, and then peripheral, and then the vimentin mean fluorescent intensity is pretty much the same across all conditions. So this means that the increase in the ECM production is due to the muscle cells. So, and then we want to check how the TGF-beta affect the structures of the bundles by staining the SAA. So you can see for the control, there's a long fibers with stratified structures, but with TGF-beta treatments, the bundles, they are disintegrated with like small pieces here. And this results in the force deductions. So we measure the twitch force, the tetanus force, here just showing the tetanus force here. And with all 3 donors, we saw a decrease in the force upon the TGF-beta treatment. And also, these treatments also lead to increase in the stiffness. So as you can see here, donor 1 and donor 3, we see a significant increase in the muscle stiffness upon the TGF-beta treatment. For the donor 2, we also see an increase, but because of the large variance in these samples, we couldn't get a statistical difference. So we next want to make sure that this is the cause of the TGF-beta. So we try to knock down the TGF-beta receptors with this drug, SB52534. It's a potent inhibitor of the type 1 receptor, AKL5, which is the receptors of TGF-beta. With this drug, we find that if we include in the inhibitor, we can restore the muscle bundle structures here. And also, we have a little bit decrease of the collagen 1 expression of the muscles, and we have an increase in the force in this donut. So this altogether shows us that we can recreate the fibrosis models with our healthy muscle bundles. Next, we want to integrate these bundles with the vessels we have to examine the functional changes due to the inflammations and the fibrosis. The hypothesis we have is the skeletal muscle fibrosis might influence the blood vessel by reducing their functions and increasing the inflammatory and fibrosis markers in both of the systems. In order to do that, we first design a small chip to hold two pairs of the muscle together, and there's an inlet and outlet. We can put them into our perfusion loop with the blood vessels here. And then we make these two systems separately at first. So for the bundles, I just discussed the timeline before. For the blood vessels, we give them 10 days for the vessels to be mature, the endothelial cell to become fluent, and then we co-culture them, integrate them for 4 days. At this stage, we withdraw the TGF-beta, so there's no TGF-beta in these 4 days. After 4 days of integration, we perform endpoint testing, including the contractile force, the blood vessel functions, the gene expressions, and the immunofluorescence for protein levels, expression levels. So we first look at the muscle, and we found that with the integration systems, we see a very similar effect on the TGF-beta treatment. It reduced the force compared to the controls. And also, even we withdraw the TGF-beta for 4 days from the system, and we see that this integration of the myo bundles persists even with the withdrawal with the TGF-beta. And also, and then we want to look at the blood vessel functions. The fibrosis muscle also affect the functions of the blood vessels. So one of the key functions we look at is the vasoactivity of the blood vessels. With the vasoactivity, we use the phenylephrine to create to induce the vasoconstriction, and the acetylcholine to induce the dilations. With the longevity muscles, the healthy muscles integrated with the blood vessels, at day 10, which is the day that they are being integrated together, we see a nice dilation and constriction, and this is pretty similar after 4 days. The case is not the same with the fibrotic muscles. We can see reductions in the dilation and the constriction. So which means the fibrotic muscle indeed affect the blood vessel functions. And also we check the endothelial cell mesenchymal transition markers, the alpha SMA, the vimentin, and the fibronectin. We saw a significant increase in the SMA and the vimentin. Although the fibronectin, there's some increase, but due to there's a point here at the control bundles, so give a very large variance, so we don't have a statistical difference now. So, and then this vimentin increase it's backed up by the immunostaining as well. We did see an increased amount of cells express vimentin after the TGF-beta treatment. And also, when we integrate the systems, the fibrosis muscle also lead to the increase of weekend staining, which is a marker of the endothelial cell activation in the blood vessels. And we quantify it by counting the VCAM1 positive cells. So as a conclusion here, we developed a human skeletal muscle fibrosis model with overexpression of the TGF-beta1. This will lead to the decrease in the muscle contractility, increase of the bundle stiffness, disintegration of the myofibers, and increase of the ECM productions. And when we inhibit the TGF-beta1 receptors, we restore the muscle contractility, and the ECM deposition is decreased. When we integrate these two systems together, we saw that the fibrotic skeletal muscle leads to the decrease in the blood vessel function in terms of the vasoactivity, and lead to vessel inflammation through the weekend expressions, and also there's a markers, expression of endothelial cell mesenchymal transition markers, vimentin and smooth muscle ethylene in our system. With this, I want to thank all the members in the Trustkey Lab and also our funding agencies, and I can take any questions here. Thank you.